Hello, my name is Dan Ricciuto from Oak Ridge National Lab. I'll be talking today about some of the modeling work that we're doing on the SPRUCE experiment, specifically at how we're confronting some of the models that we're working with observations. SPRUCE is the spruce and peatland responses to environmental change. It's a large-scale, long-term experiment, and it's assessing the responses of a northern peatland. This is a high-carbon ecosystem containing lots of carbon in the peat and below the surface, looking at the responses of that to whole ecosystem warming and elevated atmospheric CO2 concentrations. Spruce is located on a bog called the S1 bog. It's part of the United States Forest Service experimental forest in north central Minnesota. You can see on the map where it's located in the northern part of the state there, and then more broadly as to where it sits in kind of the boreal zone of North America at the southern edge of where a lot of these wetlands are located. And the types of vegetation there, primarily it consists of black spruce and large trees. This area was strip cut and harvested in 1974, and it's been regrowing since then. Below the canopy, there are also a significant component of shrubs and forbs or grasses. Also, essentially a carpet of sphagnum moss, a very mossy surface, a very wet surface. And below the surface, there is approximately 250 kilograms per square meter of carbon contained in the peat, which is a far greater amount of carbon below the ground than near the surface. This is what our experimental site looks like. We have 10 open top enclosures on the ecosystem, each of them containing its own sort of mini ecosystem with spruce, large trees, shrubs, and moss at warming levels between ambient all the way up to 9 degrees Celsius over the ambient temperature. So significant level of warming beyond what we see in the normal ecosystem. Half of these chambers also include elevated carbon dioxide concentrations at a level of about 900 parts per million or 500 parts per million above present day levels. So here's the view looking from above down into one of these spruce enclosures, and you'll see inside we have in general about a dozen trees. You can see the moss layer that makes up much of the surface, and in the center a tower where many of the measurements are made. So you can see the schematic here on the right. So the warming is accomplished above ground using a propane heating system. The heat is blown into the enclosures, and it creates a sort of turbulent eddy that cycles throughout the air volume of the chamber. And below ground, there's an electric heating system, so heating the peat to a similar level as the air, about 9 degrees in the most extreme case, warmer than the surrounding ecosystem. This slide shows the overall timeline of the spruce experiment. It's about all the way back in 2009. They began to do some pre-treatment characterization of the ecosystem, identifying the specific locations in which the experimental systems would be constructed, and that construction began in 2012. Before the full enclosures were built, the deep peat heating elements were in place, and so in the year 2014 they conducted a deep peat heating experiment looking at the impacts of warming on peat decomposition and chemistry. And then in the summer of 2015 the whole ecosystem warming began, and in the next year in 2016 the elevated CO2 part of the experiment began, and that whole experiment is envisioned to continue for about 10 years. And so here now in 2020, we're about halfway through the treatments. We're starting to see some very interesting responses, and in the next 5 years, I'm sure we'll continue to see many more interesting responses. And then the idea is at the end of the experiment, we'll be doing some more destructive type harvesting to assess how the treatments impacted the overall ecosystem. So here are some of the overarching science questions that we're interested in addressing with this experiment. One of the primary motivations for this experiment was that we knew there's a lot of carbon contained in these peatlands, and we want to know if they're vulnerable, and if so, how vulnerable to the types of atmospheric and climatic changes that we might be seeing over the next century. So specifically, we want to know and understand at what rate will ancient carbon be released from accumulated peat. Some of this peat has been building for thousands of years and is very old. So in response to deep below ground warming, are we going to see some release of that peat? And if so, what's the relative release that comes out as carbon dioxide compared with methane? So that's important because the atmospheric warming potential of methane is much higher than that of CO2, and so what comes out, that ratio is important in determining the feedbacks to global warming from these types of systems. We're also interested in what are the interactions between ecosystem responses to warming and the availability of nutrients and water. So as we warm up these systems, we may see them begin to dry out. The moss, for example, may be very susceptible to drying. On the other hand, we might see with warming increased availability of nutrients, which might help increase the productivity of certain vegetation types. So this diagram shows specifically some of the initial hypotheses that we had regarding various aspects of the system when we began the experiment. So on the left, we'll see what we thought the warming might do, for example, to sphagnum mosses. And so with warming, we hypothesized that we might see reductions in gross primary productivity and net primary productivity from the impacts of shading. So if other plant functional types such as shrubs or trees begin to grow more rapidly, there's less light available for the sphagnum, which grows near the surface. We also might see drying, which would also make the sphagnum mosses less productive, whereas in the vascular plants, we might see increases in photosynthesis and respiration from the warming, from longer growing seasons that's caused by the warming, from an increased availability of nutrients such as nitrogen, changes potentially in mortality. And then on the bottom left there, we see the potential hypothesized impacts on biogeochemistry. For example, we think decreasing water table and increasing temperatures might increase nutrient availability, might increase methane production and consumption. On the right-hand side, we see some of the hypothesized impacts of elevated carbon dioxide concentrations. More CO2 generally means more food for all types of plants to grow, and so we might expect increased productivity from all of the vegetation types. However, when we give the plants a lot of CO2, they may run into limitations in terms of nutrients. They may not have enough nutrients from the soil to grow to their maximum kind of potential. And so we may see more nutrient limitation on that side. One interesting point we had at the beginning was we wanted to understand whether different models, so models in general, produce their own hypotheses of how they think the systems will respond to these kinds of treatments. And we wanted to understand whether different models would agree with each other or with what some of the field experts might say. And we wanted to use models, Earth system models and other models to guide measurements that we might place optimally to reduce uncertainty in the model predictions. So here at Oak Ridge, we're using a model called E3SM. It's the Energy Exascale Earth System model, which is funded by the Department of Energy, and more specifically ELM, which is the land model component of that full Earth System model. So it's very similar in a lot of ways to the Community Land Model, version 4.5. We branched off from there several years ago. So compared to some of the models you may be looking at here as part of the training, this model is quite expensive to evaluate. It takes several hours to run a single site, kind of a single processor, and a couple of days if we're wanting to do a global run, generally on larger clusters with thousands of processors. And so this model includes pretty detailed hydrology in terms of representing various processes, above ground evaporation from the surface, from the canopy, and of course transpiration from the vegetation. And then below ground, it's representing 10 different soil layers that each track soil moisture and vertical flows of water, as well as the water table and the location of that, which is very important for the spruce site. It also tracks carbon and nutrients, including nitrogen and phosphorus, and the cycling of those both through the vegetation and through the soil. This slide shows specifically some of the modeling efforts that we're undertaking at SPRUCE using the ELM framework. And so we brought in representation of the microtopography that we see at the site. And so it has these hummocks and hollows throughout the landscape that can be represented, and they have important impacts on the hydrology of the site. We're also modeling 4 different plant functional types, which I mentioned, these 2 main types of trees, picea or black spruce, larix or larch. Shrubs, there are several types of shrubs present at the site. We've aggregated them together into one plant functional type, as well as the moss. In this modeling framework, the 4 plant functional types do not compete for light or radiation. In reality, they do, but this model doesn't represent that. But they do compete below ground for water and nutrients. We also have introduced a more detailed representation of methane cycling at the site. So model data integration is a really key part of the SPRUCE experiment. So we have the ELM SPRUCE model that I just discussed, but also the TICO model, which you've already heard about at this workshop. Both models are doing formal types of calibration with data, with SPRUCE data, to improve the models. Other modeling groups are beginning to participate as well. You'll hear also shortly about the EcoPad system, which can assimilate data in your real time to produce ecological forecasts at the SPRUCE site or at other sites as well. So some of the main goals of those exercises are to estimate model parameters. These models generally contain lots of uncertain parameters, and using data to constrain or calibrate those parameters can really help to improve the predictions, but also I think equally important is to provide reasonable estimates of prediction uncertainty. So how confident are we of the predictions that the models are making? So this is a framework that brings together the model code, prior information that we might have from literature or other sources about the vegetation and soil parameters, as well as forcings, or generally the atmospheric sorts of inputs that are needed to drive the model. And that, in combination with the observations that are being made at SPRUCE, can be used to do this parameter estimation step and produce predictions with uncertainty. So here's an example where we did some optimization of ELM parameters against some pretreatment biomass information that we had at the site. So in this case, we used 36 model parameters across the 4 different plant functional types involving allocation, leaf and root traits, phenology, autotrophic respiration, and stomatal conductance parameters. And all of these runs use the same initial conditions in 1974, which is the date of the harvest that occurred at the site. And so we ran genetic algorithm type of optimization where we allow these 36 parameters to vary within specified ranges and eventually reach an optimal value where the model best matched the observed biomass values of the trees, shrub, and sphagnum. And so you can see the model curves there for the trees compared to the observations, as well as the green for the sphagnum and for the shrubs as well. And so we do, after this optimization, see quite good agreement with the observations. And so by changing, optimizing parameters, we are able to get a good fit where we see, if we compare this to the original or default version of ELM, which isn't shown here, We see much slower growth than we would in an upland system, for example, and that's achieved through lower allocation of carbon to stems and higher respiration temperature sensitivity of autotrophic respiration. So on this figure, then, we can see what happens when we take these optimized parameters in ELM and run the model forward to predict vegetation under treatment conditions. And so what we're looking at here is a relative change from control for the +9, or the most extreme warming case with ambient carbon dioxide. So what we see is, beginning at the year 0 when the treatment begins, you can almost immediately see the spruce or picea there declining. So the biomass in the +9 declining relative to the control, and by year 10 it's about 70% lower than the control. And so the spruce trees with these optimized parameters that were optimized against pretreatment biomass, the model spruce tree responds very negatively to warming. Whereas with the other plant functional types, we see modest increases in larch or larix and the shrub types. There's a clear seasonal pattern there as well. For the sphagnum, there the solid green shows what's happening on the hummocks, and the dashed line shows what's going on the hollows. And so on the hummocks, we can see that that pattern is kind of closely following the water table height in red. And so these hummocks are a little bit elevated from the surface, and that means they tend to dry out faster than the hollows, which stay quite wet through the season. So that sphagnum on the hummock top begins to dry out. And so under dry conditions, you can see negative changes in biomass relative to control, but when it's quite wet, or when the water table is higher, you see the biomass responding positively. So there's a very strong hydrologic feedback that we're seeing there. So more recently, there's been an attempt to synthesize a lot of the observational data together from the first 3 years of the study and look at the response of the overall net carbon exchange to warming. And so we can see that data estimate in the red circles as a function of mean annual temperature. And there's a very strong observed response that's being seen at the site. So as the temperature warms, negative carbon exchange in this context means more carbon released to the atmosphere. And if you compute a slope of that, it's about 30 grams carbon per meter squared per degree of warming. And that's a quite strong response that we're seeing at this site. So when we take our ELM spruce model and we put the same treatment conditions in the model, we're seeing a very similar response with also a release of carbon to the atmosphere. And so the model in this case was able to reproduce the carbon loss quite well. In the model's case, primarily by looking at the moisture sensitivity of respiration. And so as the water table is dropping and drying out in those warmest treatments, we see increased respiration and more release of carbon to the atmosphere. But if we begin to drill down into various aspects of different vegetation types and their responses to warming, we still do have quite a bit of work to do. There's some key differences that remain in individual parts of the carbon budget. And so, for example, we're seeing a lot of cases in those warmest plots, the sphagnum moss is dying and basically disappearing, whereas in the model it's a much more modest decline or even stable fluxes of carbon that we're seeing into the moss. One thing that's been really valuable for us in the experiment is looking at sensitivities of model parameters in ELM and other models as well. And so we can use, when we're doing these tests and parameter optimizations, we can run model ensembles. For example, we took those same 36 parameters that we used for optimization and we ran an ensemble of ELM spruce simulations. And from that, from the variance that we see in the outputs, we can begin to understand the influences of specific parameters on specific outputs. And so this sensitivity analysis shows, for example, the first variable there, gross primary productivity, or GPP, the relative influences of individual parameters on that output. And so on the bottom, the blue is the FLNR, which stands for fraction of leaf nitrogen in Rubisco. It's a strong control on VC max, or photosynthetic uptake. And BENT there stands for the plant functional type, so that refers to the spruce trees. And so that spruce photosynthetic capacity is quite important for GPP. And we can also see this MP underscore BENT, so that's the model conductance slope parameter, which is also quite important. And closer to the top there, we see kind of across the board this particular parameter, the base rate underscore MR, that controls the maintenance respiration, which is a key component of autotrophic respiration for the site. And so that parameter tends to be quite important, and we can see with the next variable here, net primary productivity, since that includes the influences of autotrophic respiration, has a much stronger influence on that variable. So by understanding which parameters or sorts of plant traits drive the variability in these outputs, that gives us some idea of what we might need to go out and measure more carefully, or processes that we might need to understand in more detail at this experiment. One thing I think it's important to recognize is we don't really need to run the full ELM or land surface model for every kind of observation that we want to compare against. And in some cases, there may be just one particular equation or algorithm that we're interested in that we can compare that model and observations directly. And so in this case, we're looking at leaf respiration as a function of temperature. So something that came out early in the leaf level observations from the site is that spruce trees are highly sensitive to these temperature response functions. And so Tony King here has taken a number of different representations or possible representations of temperature sensitivity and fit the parameters in those models to the observations. And so by doing that, we can understand potentially which model structure is the best one to apply to these data. So we're still in the ELM using the kind of default model, and with additional field observations being collected this season, we'll be able to get a much more conclusive determination of which one of these temperature response models might be the best one to use and replace the existing one in ELM. For these types of analyses, generally the models, these kind of functional models, are much faster to evaluate than the full ELM, and so we can perform parameter estimations on them like Markov chain Monte Carlo directly, and it doesn't take very long. And so we can take those model units and calibrate parameters in them, and then bring those parameters back into ELM. We're doing a similar kind of optimization exercise for the methane model that's part of ELM SPRUCE, and so we have some detailed observations of methane fluxes at the site, which we've used to optimize 20 parameters in the model, and we're doing a relatively good job of representing the observed seasonal cycles with the model, and also looking at concentration profiles of dissolved organic carbon on the right hand side here as we look as a function of depth. So I'll wrap up by talking a little bit about what we have in mind, or what questions we have to address in the future. And one question that we're always dealing with is how much complexity or process realism do we need, or is good enough for predicting the bog-net carbon balance? So do we need a model with the level of process detail that ELM has, or can we get away with simpler carbon cycle models that may be much faster to evaluate and allow us to explore more aspects of uncertainty in the system? Another question that we deal with at this site is how much pretreatment conditions affect the regressions over the treatments. And so this is a regression-based design. We have 10 treatments and 5 different temperature levels, and so what we're really seeing, or what we're getting from the experiment, are response functions of different variables as a function of temperature. And so those might be influenced by variations in the number of trees, for example, or the leaf area index of shrubs, for example. We're not considering those things in the model now, but that may be something that's very important we need to consider in the future. Informing models from measurements across scales is a very important question that we struggle with in a lot of cases. And so we have some measurements that reflect what's going on in the entire enclosure. We have other measurements that look at very small parts of that enclosure. For example, a specific leaf, or a measurement that may be done, or a very small sort of chamber to measure respiration within the broader enclosure. So when we take a measurement from such a small scale, does it apply to the rest of the enclosure? Are there other sources of variability that need to be considered, and scaling relationships that we need to understand? And then once we have the data from the experiment, we're not just interested in this one specific site, but we're interested in its relevance over much larger wetland regions. And so these systems, similar sorts of systems, exist, for example, throughout Canada and also in Eurasia and Siberia. And so what we learned from this experiment, is that relevant for other areas as well? Are we going to see large releases of carbon and methane at other sites? Or are they so different that what we learned at SPRUCE doesn't have much relevance elsewhere? Obviously, we hope it does, but we need to use the models in a way to help us do that scaling, to take what we've learned, for example, to assimilate and calibrate parameters, and then use those parameters at other locations and evaluate them with data from other sites. This accelerated cycle of feedback, for example, the ecological forecasting that we have in mind, we hope will provide very useful information for the field scientists to plan their campaign. With other experiments in the past, what's often happened is that the model intercomparisons or evaluations are done only at the end of the experiment, and I think that's shifting now. Do we want to get the models engaged very early on so they can provide sorts of initial predictions, and we can understand how these models improve as the data come in from the experiments? And so we're seeing, I think, a much tighter coupling between the model and the experimental side. So thanks for listening in, and if you have any questions, please feel free to ask. Thank you.